All right. Well, it's 3 o'clock, everyone, Eastern time, by my count at least. So I figured I'd get started on time and reward those folks who hopped on the call. My name is Charlie Leonard. I'm a faculty member here at the University of Pennsylvania in the Center for Clinical Epidemiology and Biostatistics and the Center for Pharmacoepidemiology Research and Training. And it's my pleasure today to present the second of a series of 3 webinars focused on the health effects of drug-drug interactions. For those who were on the call last week, Sean Hennessy presented broadly on methods to study the health effects of medications, whereas today I'm going to drill down a little more and focus on methods to study the health effects of drug-drug interactions. For those of you that have registered or still wish to register, we have one final session coming up the same time next week, that is on June 29th, looking specifically at a clinical important drug-drug interaction study that we conducted. So as I pointed out today, I'll be giving a brief overview of some methods to study the health effects of drug-drug interactions. By way of disclosures, the bulk of the work that I'll be discussing today has been supported by the NIA and NIDDK from the National Institutes of Health, and I have no conflicts of interest to disclose. So here's a roadmap for our talk over the next 45 minutes or so. I'll open the floor up for questions at the end of those 45 minutes. I'll be happy to take those via chat box at that time. But for the next 45 minutes or so, I'll be talking about why we should consider conducting pharmacoepi studies of drug-drug interactions. I'll then talk about methods considerations for hypothesis-driven DDI studies, then talk about whether or not we need a priori hypotheses and can we develop and employ hypothesis-free screening approaches to examine DDIs. And then finally, I'll leave you with a little teaser of how we can ensure our findings affect clinical care. And that's really meant to be a segue into the final session of our webinar, which is next week. So I'm sure many folks on the call have seen this Chicago Tribune piece. This was an investigative reporter or group of reporters that took prescriptions for known, quote-unquote, known interacting drugs to pharmacies around the Chicago area and tried to see if they could get them filled, despite the fact that there's some evidence that they can have adverse effects on patients. So my initial reactions here was, wow, this is potentially really embarrassing for pharmacies. But, then I sat back and thought, well, I wonder what the clinical outcome data are underlying these target DDI pairs. But I may have been the only person in the world that saw this and thought that. But anyway, the point is that DDIs are in the public eye. They matter to patients, to pharmacists, to physicians, and other healthcare providers. So probably not a surprise slide for folks on the call, but DDIs are really, really important. We as Americans love medicating. As the population of the U.S. ages, polypharmacy is increasing, and this is a common phenomenon in older adults in which known drug-drug interactions constitute about 13% of all adverse drug events and up to 5% of hospitalizations. And these figures are surely gross underestimates as they only include well-documented drug-drug interactions. But despite the importance, we know incredibly little on drug-drug interactions. And probably the issue here is scale. At least according to some estimates, there are over 100,000 two-drug DDIs or drug-drug interaction pairs that have been hypothesized. And the mechanisms of those DDIs and the clinical importance of them has been vastly understudied. So not surprisingly, case reports have dominated the literature. Case reports, while useful for hypothesis generation, tend to provide a low level of causal evidence between an exposure and an outcome. Therefore, it's not terribly surprising that major DDI compendia disagree with one another. This causes confusion, perceived unimportance of particular drug-drug interactions, and ultimately the overriding of alerts that are programmed into computerized decision support systems. So given this, in 2014, the U.S. Department of Health and Human Services prioritized the reduction of patient harms from drugs, one component of which was related to drug-drug interactions. this in their National Action Plan for ADE Prevention. There was really a four-pronged approach that was targeted at surveillance, prevention, research, and incentives. And there was a major focus on reducing adverse drug events from three main drug classes, that is bleeding from anticoagulants, hypoglycemia from antidiabetics, and overdose from opioids. And within the report, they talk about the importance of identifying and preventing DDIs as a crucial component for public health, and really implore folks to both find and close existing knowledge gaps. So what would help really close these knowledge gaps is a translational approach to science. And what do I mean by translational approach? I mean passing hypotheses and knowledge within and across settings. So in particular, just in this example here I have based on a diabetes example, you can see in the left-hand panel different types of research you could think of that could explore potential drug-drug interactions, whether that be in vitro studies conducted in labs, in vivo studies, simulation work, data mining or screening approaches, pharmacoepi studies to build up an evidence base, a real-world evidence base, and then finally the development and evaluation of risk mitigation approaches. And normally folks who work in these areas tend to work in their very specific area, and unfortunately there's not lots of crosstalk in these areas. And there's often not lots of crosstalk between researchers and people in clinical care settings. And I think we would all be better served if we could help each other understand each other's expertise in science and come up with a collaborative translational approach to kind of break down the barriers to exploring DDIs. So related to that is the fact that it's well known that these silos that we operate in our specific research areas, impede the translation of scientific findings. And this has really been a structural issue that's long plagued biomedical research. And I'm not going to discuss them today, but there are many forces that really keep us inwardly focused and hyper-specialized in our area. So, when you read in the literature the fact that we should be imploding silos and all working together, well, I think that should be a goal. Really, implosion may be unrealistic. So I'd argue that at the very least, all we can do is lean on each other, learn from each other, and at least try to work across a couple different areas to improve our knowledge of drug exposures and outcomes and the health effects of medications and DDIs. So hopefully you see that there is a clear need for translational research on drug-drug interactions. We must contribute to the broader knowledge about drugs involved in these DDI pairs by screening for previously unanticipated DDIs, elucidating biologic pathways that impact pharmacodynamics and kinetics, to both predict and examine effects on surrogate and clinical endpoints, and then finally to develop and evaluate approaches to manage risk in clinical settings. Now I say we must contribute to this knowledge. Who are we? I see we as basic and clinical pharmacologists, biomedical informaticists, pharmacoepidemiologists, CDS experts, compendia editors, many of the folks on the phone now. So the bottom line is the science of drug interactions requires integrated research by a wide range of investigators. We need critical, rigorous information on mechanism, the clinical importance of the outcomes, and optimal management. And really the latter requires knowing much more than simply if two drugs interact, so the research can't really just stop there. So hopefully that's kind of primed the scene here for why conducting pharmacoepi studies of DDIs is important. I now want to talk briefly about methods considerations for drug-drug interaction studies when we have a specific hypothesis in mind. And I'd like to start by focusing on this case report. So as I mentioned earlier, case reports, while not very useful for determining cause, can be useful for hypothesis generation. So here's a case report that was published in 1991 by Ahmad about a 53-year-old woman with diabetes who had been taking a sulfonylurea glyburide for 9 months and everything was going well. She was then prescribed gemfibrozil for her high triglycerides and seen 3 weeks later in clinic. Her blood glucose was low and her husband had told the physician that she had been having attacks of hypoglycemia since gemfibrozil was started and had even fallen. So the author of this case report stated that when gemfibrozil is given to a patient receiving a sulfonylurea, that patients should be watched closely for loss of glucose control. Now, as you might have been thinking as I went through that little case, well, what's the underlying putative mechanism here? That would help us understand what might be going on. And this is a very simplistic representation of potentially what could be going on, whereby the patient ingests glyburide, there's some process of uptake and transport of the drug such that it gets into systemic circulation. And then the glyburide is metabolized in the liver by hepatic enzymes, in particular the cytochrome P450 system, maybe 3 different isozymes, primarily 3A, but also 2C9 and 2C8. And the glyburide in its active form is thereby converted to mostly inactive metabolites and then excreted. But you can imagine in this potential situation that the gemfibrozil that was prescribed can inhibit some of those isozymes in the liver, particularly the 2C9, 2C8 variants. And you can imagine a situation where inhibiting the process of converting glyburide from active to inactive could increase the concentration of glyburide, leading to an exaggerated pharmacologic effect, which leads to hypoglycemic episodes. Now, for those not familiar with DDI nomenclature, typically we would refer to a drug like glyburide in this instance as the object drug, that is, a drug that's affected in a DDI pair, whereas gemfibrozil would be the precipitant drug, that is, the drug doing the affecting. Now, investigating the clinical consequence doesn't always occur as rapidly as we would hope. So gemfibrozil was marketed in the early 80s, glyburide soon thereafter in the mid-80s. And it really took until our work in 2016 where we elucidated the clinical importance of this interaction between sulfonylurea drugs and antihyperlipidemics, including gemfibrozil, until we had some critical information on whether or not that this was a meaningful interaction. And you can see in between lots of things happened, including everyone thinking their computers were going to be fried to the introduction to the iPhone. So I'm going to talk about some pharmacoepidemiologic methods for thinking about studying drug-drug interactions. A lot of these points are well represented in a paper that my colleagues and I wrote that came out in CPT in 2016. So for those on the call that are interested in a more in-depth look at any one of these particular areas, I'd invite you to take a look at this paper. So here are some methods considerations for pharmacoepi studies of DDIs that I'll be going through over the next 15 or 20 minutes or so in this section. I'll talk about optimal research designs for DDI studies in pharmacoepi. I'll talk about the concepts of assay sensitivity in using positive and negative control DDI pairs. I'll talk about the importance of outcome validity when you're using claims data sets like we do in pharmacoepidemiology to be able to identify a valid outcome. I'll talk about the initiation order of the object and precipitant and the importance that has, as well as the time course of the interaction. So does a DDI occur early or late when concomitant therapy begins? So first with research designs. So unfortunately, there's rarely clinical equipoise for experimental design. And by experimental design, I mean the conduct of a randomized controlled trial. You know, usually we have some sort of mechanistic basis for why we think two drugs might interact. So you can imagine it might be unethical to provide one group of patients with that potentially interacting drug and another group doesn't receive it. But regardless of the equipoise issue, size and cost tend to be incredibly prohibitive for studying a clinical outcome in a randomized controlled trial. So there's heavy reliance on non-experimental or observational research, such as pharmacoepi. But what design is really optimal to use to study drug-drug interactions? You know, should we always be using a cohort study or case control study? Should we consider some of the more nuanced designs that are self-controlled designs that are case-only flavors? And I'll talk about these very briefly. So if you're on the call with Dr. Hennessy last week, He went through some general flavors of pharmacoepi study designs. He had a slide that looked very much like this, where he reminded us what the cohort framework looked like, such that we have a study population or cohort that is allocated to treated or untreated, or treated or alternatively treated by some non-random process. That is, it's not a clinical trial, so this non-random process is the physician or nurse practitioner deciding whether or not to prescribe a drug to a patient, and you follow people forward in time until they either experience or don't experience the outcome. So under the model of this cohort framework, you can imagine comparing the frequency or the rate of the outcome of interest, say hypoglycemia here in our example, in different exposure groups. So a simple approach, which is represented in this 2x2 table here, would be to measure incidence rates. That is, the number of events occurring in a certain group divided by the amount of person time that's contributed by the people in that group. So if you look at this very simple 2x2 table here, you can imagine the incidence rate of your outcome of interest, that is hypoglycemia, in kind of the base case scenario. That is, people who are neither exposed to the object drug or the precipitant drug. You could also envision what that incidence rate might be in people exposed to both of the drugs, that is in this lower right-hand cell. And then you could envision what it would be for people exposed to only the object, the incidence rate, and only the precipitant. Pardon me. And the null hypothesis would be that there's no drug interaction. That is, the incidence rate of the outcome of hypoglycemia in people taking both drugs divided by the incidence rate in the base population, that is, people taking neither drug, you would expect that to be the same as the incidence rate in people taking only the object drug over the baseline rate times people taking multiplied by people taking only the precipitant drug over the baseline rate. So if you were to reject that null hypothesis, you would say there's a drug-drug interaction. That is, a DDI would be indicative of a departure from this multiplicity in this formula. So if you look in this 2x2 table here, and these are purely hypothetical numbers that I made up, you can see that I set the baseline incidence rate in users of neither of the drugs to be, say, 0.1 hypoglycemic events per 100 person years, where in users of gemfibrozil alone, say 0.4 per 100, and in users of glyburide alone, because it's a sulfonylurea drug, higher at 1 per 100 person years. But in people taking both drugs together, that is both glyburide and gemfibrozil, you see a departure from multiplicity here, such that the incidence rate in users is 5 per 100 person years. So if you do the math, 5 divided by 0.1 is 50, and that's greater than 40. Therefore, you would reject the null hypothesis and say that maybe there's a drug-drug interaction between glyburide and gemfibrozil. But as you might be thinking, this simplistic design is flawed. So what I just showed you assumes that neither the object drug nor the precipitant drug have clinical indications that affect the incidence rate of hypoglycemia in this example, or that you know the indications and you fully measured and controlled for all the differences in those groups, which we clearly haven't done in that simple 2x2 table. So in pharmacoepidemiology, this is something that we think about, which is known as confounding by indication, that the indication for the drug can often dictate drug choice and may affect the outcome. And that is the fact that something related to both exposure, that is whether or not you get the precipitant, and the outcome could be a potential confounder, and this could cause an observed association to be different from that from the true causal effect. So approaches we take in pharmacoepidemiology, many of them are focused on minimizing this concept of confounding by indication. So one way to start to get over this is to better to create a cohort that's solely comprised of person time exposed to the object drug, or glyburide in this example, where you would compare people exposed to glyburide compared to people exposed to glyburide plus gemfibrozil. So all of the people in the study are exposed to glyburide, so they all have the same indication They may have different levels of severity of diabetes, but at least you know they all have diabetes. But this still assumes that there's no confounding by indication for needing that precipitant drug, that is gemfibrozil. And therefore, this approach assumes that the precipitant alone doesn't affect the outcome either. So another way to get around this might be to think of comparing people that are exposed to the object drug and the precipitant, comparing those to people exposed to the object drug and some other drug that's like the precipitant but might not be expected to interact. So in this example, that'd be people exposed to glyburide and gemfibrozil, the precipitate we're interested in, comparing those to people exposed to glyburide plus fenofibrate. Now, fenofibrate is a drug used for the same indication as gemfibrozil, high triglycerides. So this approach would minimize confounding by indication if that alternate drug, that is fenofibrate, is a true therapeutic alternative to gemfibrozil, which it seems to be. But it also assumes no interaction between the object drug and that alternative precipitant. People could argue that fenofibrate also could interact with glyburide, so this may not be the best alternate precipitant drug. So everything that I've talked about so far has been really looking at a cohort framework, but you could also think of drug-drug interaction studies in a case control framework where you're really turning the cohort framework on its head. And again, this was presented last week by Dr. Hennessy, whereby you have the same study population and the same non-random process, but you're identifying cases, that is people who had the event, and you're sampling among controls at the time that the case developed the event, that is incidence density sampling. So there are reasons to use case control studies, particularly when you have de novo data collection, but there are also some computational efficiencies you get when using a case control versus a cohort study. And these efficiencies, because of the complexity in programming drug-drug interaction studies, these efficiency gains can be magnified. But case control studies present their own sorts of challenges when being used in pharmacoepi, and in particular DDI pharmacoepi, is that it's more challenging to figure out when to measure for potential confounders in a case control study. So in a cohort study, you start to follow people when they start a drug of interest, and a certain amount of time before then is known as baseline time, and you're measuring confounders at that time to adjust for. And you know those confounders are occurring before exposure. In a case control study, that doesn't always necessarily happen, and there can be some concern of controlling for what are known as intermediate variables, that is, factors that are affected by exposure, thereby occurring after exposure, but you're controlling for them, and that can cause some bias. So generally, but not always, folks tend to use cohort studies because of this reason, rather than case control studies, particularly when looking at DDIs. You can also think about conducting self-controlled studies, that is, studies that include only people that have the outcome of interest, that is hypoglycemia. In these types of study designs, people serve as their own controls functionally. So this inherently controls for factors that don't change over time, whether or not they're measured or unmeasured confounders. So for instance, genetics, to the extent that genetics don't change over time, because you're comparing time within individual, basically asking why does the event happen to the individual at the time it does than some other time. You're essentially controlling for these time invariant factors. But this requires within-person variability and exposure to the precipitant while exposed to the object. That is, the design requires discordance such that the person has some time on and off of exposure to the precipitant. So these designs tend to be most useful for both acutely or episodically taken precipitant drugs, for example, antibiotics that aren't taken chronically. But there can still be an acute indication for that precipitant, which can cause some confounding by indication. And there are approaches in self-controlled studies, like in cohort studies, to control for time-varying confounding. So for those of you more interested in seeing some of these approaches applied in practice, There's a cohort study that we published in Diabetes Research and Clinical Practice, I guess last year, that uses the cohort design to look at the clinical importance of a DDI. And then also from our group a number of years before that in CPT, they utilized both the case control design and one of the self-controlled designs known as the case crossover study. So I also want to talk about the concept of assay sensitivity. And these are methods to employ in your study, whatever study design you use, particularly cohort studies, to both quantitatively and qualitatively contextualize your findings. So again, we've been talking about the scenario in which you have glyburide as the object drug, gemfibrozil as the precipitant drug, and we're looking for an outcome of serious hypoglycemia. But you can imagine, to contextualize the hazard ratio or whatever effect estimate you get out of that study, you might also want to include users of a negative control object drug, that is metformin and gemfibrozil. Metformin isn't metabolized by the liver. It's completely renally excreted. So there'd be no reason to think that the addition of gemfibrozil, which inhibits liver enzymes, would cause an increase in metformin concentration and lead to hypoglycemia. Further, metformin in and of itself is thought to rarely cause hypoglycemia. So looking at point estimates that come out of your study when using this negative control object drug can help put into context what you find when looking at your drug pair of interest. In a similar vein, you could also include users of a negative control precipitant drug, that is, users of glyburide and pravastatin rather than gemfibrozil. Pravastatin, while not a fibrate, is still used for lipid control. Pravastatin is not thought to utilize or only a small proportion inhibit the cytochrome P450 system. So you can imagine that pravastatin wouldn't cause any increases in glyburide leading to hypoglycemia. To the extent that you know one, you could also think about a positive control precipitant drug. So, for instance, if there's information in the literature that there's a well-known interaction between glyburide and some other drug, and there's an expected magnitude of effect of that on hypoglycemia, you could see if you could replicate that in your data. And again, all of these things can help you contextualize what you find in your overall study, where you're looking at glyburide and gemfibrozil increasing hypoglycemia risk. So here's an example of how we've employed assay sensitivity in one of our prior studies. I'm not going to specifically go through the results of this study. This is actually part of my talk next week. But I do want to point out how we've used both negative control precipitants and negative control objects. So just to orient you to what's here, this was a cohort study looking at different object drugs, glyburide, glipizide, and glimepiride, and concomitant use with different antihyperlipidemic drugs, which are represented by each of the different shapes here, and really looked at the hazard ratio for serious hypoglycemia within 180 days. And our main drug of interest was really glyburide, but we also used metformin as our negative control object drug by way of a qualitative comparison. That is, we didn't use these numbers numerically in any way to adjust our glyburide findings, but simply eyeball them to see what does it look like when we don't expect an interaction, but there's concomitant use of these drugs. But we also used a quantitative comparator that is a negative control precipitant, such that glyburide users concomitant with atorvastatin users were compared to glyburide users concomitant with pravastatin users, because, as I mentioned before, we don't believe pravastatin to utilize, or to inhibit rather, the CYP enzyme system, so we wouldn't expect an interaction. So each of these pairwise comparisons of drug-drug interaction pairs were compared to concomitant users of that object drug with pravastatin. Also important for pharmacoepi studies of DDIs and pharmacoepi studies in general is the concept of outcome validity. So you can see here that the outcomes we're often looking at in DDI studies can be exaggerated or diminished pharmacologic effects of the object drug. That is, hypoglycemia is an exaggerated effect of glyburide. In another DDI study we've conducted of clopidogrel, which begins in the inactive form and is converted in the liver to an active form, that if you inhibit that conversion, you basically get a diminished effect of clopidogrel, which can lead to stroke. But outcome identification is critical to minimize misclassification. I won't go into this in detail, but misclassification, in particular of an outcome, can have serious effects and induce bias in either direction in epidemiologic studies. So therefore, when you're conducting pharmacoepi studies, in particular with DDIs, you're using administrative claims data or electronic medical record data. You need to have an algorithm or a method by which you're going to identify the clinically important outcome of interest. And whether or not this outcome has high validity metrics or performance measures is critical to minimizing bias in your study that's mediated by misclassification. So either what you do is you go to you do a literature search, you go to the literature and you identify validation studies that have already identified algorithms. So in particular, this study by Adit Gindi identifying an algorithm for hypoglycemia, or you have to embark on typically a complex de novo study to look at the validation of your outcome. And this involves getting ethics approval, identifying putative outcomes based on codes in your data, then going and getting medical records corresponding to those claims, reviewing and abstracting those records, having experts, in particular physicians, adjudicate those outcomes, and then reporting findings. And then after you do that, you end up quantifying the validity. So hopefully you can point to the literature and have this information at hand, but if not, you're conducting, as I said, a de novo study. But because many DDI pharmacoepi outcomes are binary, like hypoglycemia, yes or no, these are the metrics that you tend to be interested in. So sensitivities and specificities, which are known as validity metrics, in which truth is the denominator. So sensitivity is the probability of testing positive if the outcome is truly present. And by testing positive, I mean the algorithm that you've identified identifies someone as having the outcome of interest. Obviously, there's a trade-off between sensitivity and specificity. And these are critical measures when looking at whether or not to use a certain algorithm for identifying your outcome. But you also want to know information generally about predictive values. Predictive values are performance measures. They're not actually direct measures of validity, and that's because truth isn't in their denominator. Positive predictive value, for instance, is the probability that the outcome is truly present if the algorithm triggers and says it is. Predictive values depend on both the sensitivity and specificity in addition to the prevalence of the outcome. So here's a table I adapted from Epidemiology in Medicine by Hennekens, in which at a given sensitivity and specificity for an algorithm, you can see that the positive predictive value varies widely depending on what the prevalence of the outcome of interest is in that underlying population. So this is particularly important if you're going to grab an algorithm from the literature that tends to be off the shelf, especially since validation studies tend to only present positive predictive values, to understand what the underlying prevalence was of that outcome in the population under study and how that might differ from the population that you're applying it to. There's also the concept of the initiation order of the drugs in the drug-drug interaction pair, that is the object drug and the precipitant drug. So in our case that we went through earlier, this was a patient for 9 months that had been taking glyburide, and then gemfibrozil was added. So as I alluded to before, when using a cohort framework, we tend to follow people to minimize confounding by indication when they start, when they're concomitantly exposed, rather, to both the object drug and the precipitant drug. And that defines cohort entry. And then we follow people forward, normally over a pre-specified maximum follow-up period, until they either have or don't have the event of interest. So in this case, similar to as reported in this case report, the individual had been on glyburide for some time, and then their entry into the cohort was triggered by starting gemfibrozil. So in DDI pharmacoepi, we consider these types of episodes as precipitin-triggered cohort entry. That is, the addition of the precipitant to the ongoing object drug brought someone into the cohort. You could also envision the inverse scenario in which someone had been on gemfibrozil for some time, then was prescribed glyburide, and that precipitated their cohort entry, and the addition of glyburide, that is the object drug, triggered cohort entry. So these kinds of observations we tend to consider object-triggered cohort entry observations. And then the other scenario is in which both the glyburide, the object drug, and the precipitant drug, gemfibrozil, are started at the exact same time. That triggers cohort entry for a cohort study. And these scenarios are considered combination-triggered or combo-triggered cohort entry. That is, both drugs starting at the exact same time. Now, why might this matter? Well, I'll argue it's important to examine the associations of interest, that is, between your object drug and your precipitant drug with your outcome, stratified by groups with different initiation orders. And this is particularly relevant when the object drug is dose titrated. So you can see here in this first scenario, like in our case report, this individual had started on glyburide. During this time period, the physician's probably tinkering with the glyburide dose to maximize its effect to bring down blood glucose. But at some later point, gemfibrozil, which inhibits the metabolism through the CYP enzyme system, is going to inhibit the metabolism of glyburide and can cause an increase in glyburide concentration. So you can imagine in people that have this profile, that is precipitin-triggered entry, that the risk of the drug-drug interaction might be highest soon after the precipitant drug is added. That is, right when they enter the cohort in the cohort framework. In this scenario, where gemfibrozil has already been on board in a patient in whom glyburide is started later, this is an object-triggered cohort entry, you would imagine that the highest risk period might actually be at the end. That is, because glyburide is being dose titrated while the cytochrome P450 enzyme system is already inhibited, and only when you take that inhibition away does the person become at highest risk for the drug-drug interaction. And you can imagine that same scenario applying in the combo-triggered group. It's also important to think about and investigate the time course of drug-drug interaction. So even absent a DDI, the risk of an outcome or hypoglycemia alone can vary over time as disease severity changes, patients have infections and other things that can increase and decrease risk. And this is primarily the driver of the rationale for studying incident users in pharmacoepi. That is, you want to follow people from when you see them begin therapy and then follow them forward over time, rather than study a mix of people who had been on the drug for a really long time versus people newly initiating the drug because they might have differential patterns of risk. But in particular, in the drug-drug interaction context, in the presence of a precipitant during ongoing object drug therapy, you might expect soon thereafter to be a short peak in risk and then a decline. And it might look like this pattern. So you can see here on the y-axis, pretend this is the rate of an adverse event of hypoglycemia. So you can see someone started on an object drug like glyburide, and they have some baseline rate of having hypoglycemia while they're just on the glyburide alone. Now, when gemfibrozil is initiated and the CYP enzyme system is blocked, the concentration of glyburide goes up. So they might be expected to have a bump in the risk of experiencing the hypoglycemic episode. But if they don't have that, that risk might go down over time and eventually even dip below what the baseline risk is if they were on the object drug that is the glyburide alone. And you could think of a number of reasons why you might see this pattern. This could be the concept of depletion of susceptibles, such that the people that are likely to get the adverse event are likely to get it early, and if they don't get it, they might be resistant to it, so never get it. It could be a biologic adaptation to the pharmacologic effects of the object drug. It could be the body's upregulation of the affected metabolic enzyme, that is adaptation to the addition of the gemfibrozil and therefore upregulation of the CYP enzyme system. This could be dose reduction either by the provider or the patient doing it on their own, that's prompted by early signs of toxicity if the patient starts to get lightheaded or the blood glucose starts to drop too low. So you can envision this pattern happening for any number of reasons. But the problem is, if you don't consider the timing of the drug-drug interaction, if you don't consider different windows of time after someone is on both the object and precipitant drug, essentially you're looking at an average risk of the hypoglycemia over the entire follow-up period. And that would be an average of this peak and this dipping below what the baseline risk is. So you might not see an elevated effect or an elevated hazard, whereas in reality, you know that one's occurring. It's just a bump early. Therefore, it's important to examine pre-specified time windows to look at duration response. So still looking overall over some time window if there's an increased risk of hypoglycemia, but also to break that time window down into smaller windows to look for these transient increases and decreases of risk. And here's a time course example. I'll be going through this more in detail next week when I go through this drug-drug interaction in particular. But as a little bit of a spoiler, when we studied glyburide and gemfibrozil users, and we looked in different time windows, as you can see here along the bottom, you can see that the hazard of hypoglycemia was relatively consistent over these windows when gemfibrozil was the precipitant. But when fenofibrate was added, you can see that the risk of hypoglycemia didn't occur early, but rather peaked in the second month of therapy and then kind of went back to baseline over time. So had we not investigated these pre-specified time windows, it's possible we could have missed this potential increase in risk in concomitant use of glyburide and fenofibrate that seems to occur in a little bit of a delayed fashion. So everything I've talked about up until now has really been based on having an a priori hypothesis, a putative mechanism for why a drug-drug interaction might occur. But do we need a priori hypotheses at all? You know, could we develop a hypothesis-free screening approach for DDIs? And the answer is yes. So I'll argue we need a systematic approach to identify DDIs for further investigation. And while there's been a fair bit out there where there's been screening using spontaneous reports, as Dr. Hennessy talked about last week, there are limitations and reporting bias that's inherent in spontaneous reporting data. So can we take advantage of large claims data sets to use some of these pharmacoepidemiologic methods and designs to screen for potential drug-drug interactions that we'll want to follow up in later studies? And what I'll be introducing very briefly here is a collaborative effort that we here at the University of Pennsylvania have been undertaking with Indiana University to develop such an approach. So our objective of this work was to identify putative drug-drug interactions with sulfonylurea drugs and meglitidine drugs as objects, and to look for serious hypoglycemia, and to basically run through hundreds and hundreds of putative precipitant drugs to see if we could see any signals to follow up in more rigorous studies. So to me, this is a simple but important example of silos kind of collapsing on each other and taking advantage of the clinical pharmacology expertise that Indiana University has in conducting pharmacokinetic simulations and informing and putting forth potential DDIs that have a theoretical basis with our group's expertise in pharmacoepi approaches and developing pharmacoepi screening to identify DDIs with a clinical relevance. And then essentially putting that information together getting clinical experts in the room to see which one of these or which handful of these we should investigate further. Now, the information we've been getting from our pharmacology colleagues at Indiana University has been information like AUCR, or area under the curve ratio. That is, these are plots of drug plasma concentration versus time that predicts the ratio of systemic exposure to object drugs in the presence versus the absence of the precipitant. And combining that information with using self-controlled case series pharmacoepi designs in administrative data sets to develop these heat maps to look at increases in rate ratios for given object drugs, as you can see along the top here. And again, this is a high throughput method where we look at hundreds and hundreds of precipitant drugs that are concomitantly prescribed with these object drugs, and looking for signals that suggest potential harm either overall or in different time windows. So for instance, there's potential signal here of an interaction between glimepiride and bumetanide, and glimepiride and buspirone in terms of instigating a hypoglycemic event. And it's taking this screening information and not saying that this is causal between the exposure, or that is the drug-drug interaction pair, and the outcome of interest, but rather to take this information and think through a more rigorous epidemiologic study to investigate these signals further. For those interested, Eileen Hahn, a postdoc that was in our group, published recently in epidemiology on this method, along with our colleagues at Indiana University. So the bottom line is there are efficient approaches that have been developed to identify putative DDIs of clinical concerns utilizing these large administrative data sets that we have access to in the pharmacoepidemiology world. And that we can use these data and use these signals that arise from these data to drive rigorous etiologic studies like the one that I'll be talking about next week to inform clinical care. So we have additional steps that we're conducting to follow up on some of these secretagogue signals. We're also conducting screening of potential precipitants to be concerned of with warfarin use and direct-acting oral anticoagulant use that, if you keep your eye out, will be forthcoming in the next year or two. Then I just want to finish up by talking about how we can ensure findings impact clinical care. We can do that by conducting rigorous and reproducible science, by focusing on clinically meaningful endpoints, for example, serious hypoglycemia, not just transient changes, maybe a few points here or there in blood glucose, and by prioritizing dissemination activities, which was really one of the drivers for why Dr. Hennessy and I put together this webinar series. And then finally, engaging both traditional and non-traditional stakeholders in the process of drug-drug interactions and DDI research. So I'll end with a plug for, before I take questions, that is a plug for our next and final webinar of this series, which is next Thursday, where I'll be talking specifically about what we found in our study looking at sulfonylureas and lipid-lowering drugs. I do want to acknowledge my colleagues that helped with the bulk of the work that I presented today. Sean Hennessy, who's the director of Penn Center for Pharmacoepidemiology Research and Training, as well as Warren Bilker, Eileen Hahn, and Colleen Brenzinger at Penn, the late Dave Flockhart at Indiana University, Lang Lee, who's our clinical pharmacology expert at Indiana, as well as James Flory at Cornell and Josh Gagne at Harvard. And I do want to point out one last thing before I take questions, that folks who are interested in learning more about the types of research we do here and learning to be able to conduct such research, that there are pharmacoepi training opportunities at Penn. You can learn more at the website here in red, but there are different levels of training programs that range from a clinical research certificate to a master's program in clinical epidemiology, as well as a more in-depth PhD in epidemiology. So I will stop there, and I will see if folks have any questions. If they could put them in the chat box, and I'll kind of say them out loud and answer them as best I can. So first question I see here is, how frequent is it to see different effects in cohort entry scenarios? And I would say, in my experience, and probably conducting about 5 to 8 etiologic studies, maybe a little bit more, that not always, but we have seen them. And again, as a plug for joining the session next week, we indeed saw one in the study that I'll be presenting there. I think it's often dependent on whether or not the object drug is dose titrated. So, for instance, when you're looking at things like diabetes drugs or anticoagulant drugs, potentially things like digoxin. All right, so feel free to send any questions through. I'll wait a few more minutes just in case folks are typing. All right, let's see here. All right, this one's a little long. So you mentioned DDIs between 2 drugs. Do you have any comments on identifying potential interactions with 3+ drugs? So yes, we thought about this a lot. So as you can see, or hopefully glean from some of the methods, things I went through today, there are already inherent challenges in trying to adapt pharmacoepi methods to studying drug-drug interactions, that is, a 2-drug pair. Things probably get increasingly complex with 3-drug interactions. I mean, from my personal perspective, there's so little out there on the clinical importance of 2-drug interactions. I do think there's plenty for us to do there first before embarking on 3+ drug-drug, that is, drug-drug-drug interactions, although I do think it's an important area. Another comment, could you comment more about the dangers or benefits of propensity score adjustment, and what about instrumental variables? Right, so I'll talk a little bit about this in the session next week. So again, the goal is in these types of studies to minimize confounding by indication. That is, to make the 2 groups or more than 2 groups that you have under study be balanced in terms of baseline covariates, such that you can approximate what's a randomized controlled trial. Although you never get to approximate that, you utilize approaches such as propensity score adjustment or stratification, or an approach that was developed by Harvard, such as high-dimensional propensity score approaches. So in most of our studies, in order to get as complete control of measured confounders and to try to capture some proxies of unmeasured confounders, we tend to utilize empiric approaches such as the high-dimensional propensity score approach to identify covariates to include in the propensity score and adjust for in models. Instrumental variables are a little bit more challenging because it's often difficult to find an instrument. Let's see. Have you ever used pharmacoepi to study additive pharmacodynamic, not pharmacokinetic effects? So I can't think of one off the top of my head. I'm sure we've looked at things that have been more PD-based than PK. The bulk of the work we've conducted, though, has looked at drugs that have either inhibited the cytochrome P450 system or blocked transport enzymes like PGP. But I can think of no reason why you wouldn't be able to apply some of these same methods to examine pharmacodynamic drug interactions. Let's see. So I mentioned disagreement between compendia early on in the presentation because there's already a lack of evidence behind DDIs. How valuable is it for compendia to alert on theoretical interactions? Yeah, so this is a bit of a thorny subject. I know some folks have done research in drug interaction space. In particular, I think I'm aware of Dr. von Salker, who's looked at critical drug-drug interactions that should always be alerted on. And, some others have suggested that many or all other DDI alerts should be turned off. I think the best I can say at this point is pharmacoepidemiologists like Dr. Hennessy and myself, and hopefully others in the field, can continue and contribute more of an evidence base to help inform which of these drug-drug interaction alerts should be turned on versus turned off. What suggestions do you have for drug information vendors that curate DIs? I guess I would say, so from my experience, some folks that I've talked to have felt less comfortable evaluating evidence that's been generated from pharmacoepidemiologic studies like the ones that we conduct. So a lot of what's informed in some, drug information groups or in, that's used by DDI vendors has really focused on theoretical interactions and those from case reports. So I guess that's really our goal in disseminating here, is to get folks more comfortable with some methods they might see and to really make some of the papers more visible that have examined clinical effects in real-world populations. Let's see. It would seem that the same issues you raised for pharmacoepi would apply to developing predictive models for outcomes that include DDI exposure as a feature. What are your thoughts on predictive modeling versus pharmacoepi? I haven't given tons of thought to that, but now I certainly will. Just going back to see if I missed any other questions. All right, well, not seeing any new questions come through, I want to thank everyone for joining today. And hopefully for those of you that have registered or those that still wish to register, there's one final session of this webinar series, and it is next Thursday at the same time, 3 p.m. Eastern. And thanks again.